Recording started.
All right, so this is the first chapter of this third unit, which is going to be the nervous system, focus on the nervous system. So nervous tissue will be the first one, then we'll do the spinal cord, and then we'll do the brain. So this one is nervous tissue, looking at neural tissue first. We'll be looking at neurons and what are called neuroglial cells, and then we'll look at the primary physiological process, which is called neurophysiology, which is how impulses are generated and conducted along an axon to a target. So that's what we're looking at today in this chapter 12 lecture of nervous tissue. Let's get a feel for how this nervous tissue is organized, what it's made of, and how it works. So as I said, the things we're going to be looking at, the primary organs we're going to be looking at after we do this chapter is going to be the brain and the spinal cord. Those are the primary organs of the central nervous system, but there also are peripheral nerves, such as nerves coming off the spinal cord or the brain. Also, we have sensory receptors, things like the eyes and the ears, which are special sense organs, and we'll look at those later on in the semester. So to get just sort of a general background of the nervous system, so this is really our first go-around specifically with the nervous system, we want to talk about sort of the anatomical divisions and really how this whole system is organized. So we just sort of have a general working knowledge of this system and how it's set up. And so a lot of this you probably have at least some familiarity with by this point, but we'll go ahead and run through this. The two anatomical divisions of the nervous system are the central nervous system and the peripheral nervous system. Central nervous system, of course, includes the brain and the spinal cord, and the peripheral nervous system is just everything outside of the brain and the spinal cord. Right? And so the central nervous system, as I said, spinal cord and brain, which is what we'll be doing next in our lectures coming up later on in this unit. And in this central nervous system, we have nervous tissue, of course. We're going to see neurons and axons and connective tissue, blood vessels to supply nutrients and oxygen. So a lot of really important components, obviously, here in the central nervous system. And the functions of the central nervous system is essentially can be sort of generally categorized in the three sections. First, bringing in sensory data from either inside or outside the body, sending out motor commands to peripheral organs, like out to a skeletal muscle, telling a skeletal muscle to contract, for instance, which is something we looked at last time, right, when we talked about skeletal muscle. We saw the neuromuscular junction and how the nervous system interacts or interfaced with skeletal muscle. And then finally, which is sort of its own category of itself, is just sort of higher order functioning of the brain, like intelligence, memory, learning, emotion, all these types of things. So you can sort of categorize generally what the central nervous system is going to do, and bringing sensory information in, sending motor commands out, you can control things, and then finally higher order functions. So that's the central nervous system. Peripheral nervous system is, again, just any nervous tissue that's outside of the brain and the spinal cord. And the function of the peripheral nervous system, of course, then is to deliver sensory information to the central nervous system, to the brain and the spinal cord, or to carry out some sort of motor command. Right? The third category of higher order functioning and memory and emotion and those types of things, that's not done by the peripheral nervous system. That's only done in the brain. So it's either bringing the sensory information in or carrying motor command out to a peripheral target is what we're going to see with the peripheral nervous system. These are nerves, right? Nerves that emanate off of your spinal cord and your brain that go throughout our body to targets. Right? So nerves, as I said, peripheral nerves, bundles of axons that carry information. There are cranial nerves. You guys get to learn all the 12 cranial nerves that attach to the brain. And then spinal nerves. We'll talk a little bit about those in the next chapter when we do the spinal cord. So you can also divide this peripheral nervous system functionally into the afferent and efferent divisions, right? Afferent with an A, efferent with an E, right? To help you remember that. But basically, it's what I just described, right? So instead of saying bringing sensory information in, right, to the central nervous system, we're calling that the efferent pathway with an A, right? Instead of carrying out motor commands, we're calling that pathway the efferent division. So one way to help you remember this is E, like having an effect, right? That's what it's going out to, have an effect at a target. So those are carrying out motor commands from the central nervous system to the peripheral nervous system, muscles and glands that it's going to target. Okay? So receptors and effectors are different, right? Receptors are going to bring information in, like eye and the ear, or just even generalized sensory receptors like in our fingertips, for instance. That's going to bring information into the central nervous system. And then an effector is going to send out an appropriate response out to our skeletal muscle or to our glands or whatever target those are going to. Okay? So this efferent division with an E, like I was talking about before, can be actually subdivided into two parts. You have what's called the somatic and the autonomic nervous system. So if you take this, how you can target things and how you can control things, you have somatic and autonomic. And so somatic is exclusively associated with messages going to skeletal muscle. Okay? The autonomic division, right, autonomic nervous system, or ANS, which we're going to spend the whole chapter on later on in the semester, controls pretty much everything else. The autonomic nervous system controls subconscious actions of smooth muscle, cardiac muscle, and glandular secretions, right? So subconscious is a really important part of that, right? And that autonomic nervous system can even be subdivided further into what's called the sympathetic and parasympathetic. And you may be familiar with this, but again, we're going to spend plenty of time on sympathetic and parasympathetic. When we do the autonomic nervous system, the sympathetic typically is your sort of fight or flight type of response. Parasympathetic is your rest and digest, as it is referred to. So again, we'll talk more about that, but just sort of have a general understanding of how this nervous system is organized based on those sort of points that we mentioned. All right, so now that we have a general background of how the nervous system is organized, we can now actually start to look at the tissue, neural tissue in the body, and specifically the two types of cells you're going to see in nervous tissue. Two types of cells you're going to see are neurons and neuroglia, or glial cells as I'll refer to them. Neurons are the parts of the nervous system that send and receive signals, right? Communicate messages. That's really the primary sort of functional purpose of the nervous system, right? If you want your skeletal muscle to contract, it's going to start with a message in your brain typically or in your spinal cord and go out to the muscle, and the muscle will then contract, right? So it's cells that send and receive signals. Glial cells have nothing to do with sending a message or communicating a message, right? Glial cells are a group of cells, different types of cells, that are designed to support and protect neurons, so sort of allow neurons to do what they do best, which of course is communicate messages. So glial cells and neurons are what we're going to be looking at. So the first thing we'll look at then, of course, are the neurons. The neurons, again, as I said, this is the basic functional unit of the nervous system. This is what communicates a message, okay? And there are different types of neurons, as you probably might guess. One we're going to look at is what's called a multipolar neuron. It's very common in the central nervous system. Multipolar neuron has a cell body, has dendrites typically extending off of the cell body, and then a long single axon, which is like a power cord, right, that sort of conducts the impulse out to the target, right? If it has to go from a neuron in your brain down to a muscle in your quadricep, right, that's a long way it has to get there. So it has to travel through the cerebrum, down the brainstem to the spinal cord, and then out a peripheral nerve all the way out to the muscle. And that's usually conducted on an axon. Okay? So you can see in this image the cell body, which has a nucleus, has dendrites extending off of it, and then it has this long axon, which ends in telodendria and then communicates to a target. It could be another nerve cell, it could be a muscle cell, it could be a gland, whatever that message is getting sent. Is traveling along that axon to the target. All right. All right. So the cell body is the largest, contains a large nucleus and nucleolus. Also has cytoplasmic contents referred to as percaryon. The mitochondria, of course, are in the cell body that help produce ATP energy for the neuron. There's also rough endoplasmic reticulum and ribosomes which help manufacture neurotransmitters. So a lot of things going on in the cell body, like the cytoskeleton producing neurofilaments and neurofibrils, also Nissl bodies, which are dense areas of these rough endoplasmic reticulum and ribosomes. And so as a result of all these components, these neurofilaments and the Nissl bodies and the nucleus and the mitochondria, all these things together are going to make nervous tissue appear gray, or what we refer to as gray matter. So like, for instance, if you look at a cross-section of the brain, do like a frontal section of the brain, you will see like gray matter at the top, white matter in the middle, gray matter towards even deeper. And so the body, the nervous tissue is divided up, whether you're looking at nervous neuron cell bodies or axon. And the neuron cell bodies are the gray matter, or the darker areas of the neural tissue. Okay? Dendrites are highly branched, dendritic spines extending off of the neuron, can receive information from other neurons to extend the communication. And then coming off of the neuron itself is an axon. Axon is this long, cord, for lack of a better term, that conducts or carries the electrical signal or the action potential to the target. Okay? So the axon is really essential in this process. The axoplasm and axolemma, the membrane that helps conduct this impulse. The axon hillock and the initial segment are the initial parts of the axon where oftentimes the impulse is initiated. The axon will branch into what are called collaterals, and then ultimately telodendria, which are fine extensions of that axon, and they will end, which we've talked about before, at what's called a synaptic terminal. Right? These are the tips of the telodendria where communication with the target occurs. And so you can see that is a great image, or you can see the neuron cell body, of course, up here. You can see all the components of the mitochondria and the Nissl bodies and the neuron cell body and all those things that are giving it sort of a darker, grainier appearance. But then you have this axon that's going to conduct the impulse towards the target. In this image, it looks like it's another nerve cell, but again, it could be a muscle or a gland, cardiac muscle, whatever, smooth muscle, whatever the message is getting communicated to. All right? And so it ends at a synapse, right? The synapse is the site of communication with the target. And we saw a synapse when we looked at skeletal muscle last time, right? This was what we referred to as the neuromuscular junction, right? The coming together of the nervous system with the muscular system. And so that's what a synapse is. Structure of a synapse is you have a presynaptic cell, that's the neuron that's sending the message. The postsynaptic cell is what's receiving the message. And the synaptic cleft is the small gap or space between the two, right? And so they're not connected. Most times this communication is done through chemicals, right? Chemical moves across the space and binds to receptors at the postsynaptic cell and then has its effect, all right? So, as I said, this expanded area contains neurotransmitter, and the neurotransmitter are the chemical messengers that communicate the message, right? They can affect receptors of the postsynaptic membrane, be broken down by enzymes and reassembled. And so, for instance, ACH, right? That's one that we have seen already. Acetylcholine is one example of a neurotransmitter. There are many others: dopamine, serotonin, epi, norepinephrine, these types of things we'll talk about later as we get more specific. There are many types of neurotransmitters, but these are what we call chemical messengers. And so here's a nice shot where you can see the neuron cell body, the telodendron ending at the synaptic terminal, has the mitochondria and the synaptic vesicles containing the neurotransmitter, and then here's the postsynaptic target. All right, so neurons can be classified structurally. As I said, there are different types. Not all neurons are exactly the same. Okay? So you have bipolar and unipolar and multipolar neurons, as I mentioned before, and you can see images of all these types. Most common, the one that we'll refer to most frequently, although we may mention some of these other ones, but the most common one that we'll look at is what's called a multipolar neuron. We have the cell body at one end, a long axon ending at a synaptic terminal. All right, so neurons can also be classified functionally, okay? And you have, sensory neurons, of course, that are bringing sensor information into the central nervous system. You have motor neurons, of course, that are sending information out to the body, right? Commands and things like that. But then, and those we've sort of talked about a little bit already, but then you also have this thing called interneurons. Interneurons are basically neurons, association neurons that stay within the central nervous system and sort of communicate and conduct messages to other neurons within the central nervous system. So they're not coming or going, so to speak, right? They're just staying within and maybe amplifying a signal or communicating a message, making a reflex more complex, things like that is what interneurons are responsible for doing. All right, so sensory neurons, again, these are bringing sensory information into the central nervous system. Typically, they're unipolar, and again, they're going to either monitor the internal environment, we call those visceral sensory neurons. They can monitor the external environment, we call those somatic sensory neurons. But ultimately, they're bringing information about the either outside world or world inside our bodies to the central nervous system from the peripheral. And of course, this is coming in. So this is the afferent A, with an A, sending information in to the central nervous system. 3 types of sensory receptors: interoceptors, exteroceptors, and then what we call proprioceptors. Interoceptors, of course, are monitoring the internal systems of the body, like digestive, cardiovascular, etc. Exteroceptors are responding to external senses like touch, temperature, sight, and proprioceptors are monitoring position and movement. Okay? Motor neurons carry instructions from the central nervous system to peripheral, right? So sensory neurons bring information in, motor neurons carry instructions back out, and so these are efferent with an E, right, having an effect to a target, right? And so there is either, we already talked about this, but there's somatic motor neurons or they're autonomic motor neurons, right? Somatic are going to skeletal muscle only, and autonomic going to everything else, smooth muscle, cardiac muscle, glands, etc. All right? And then finally, interneurons. Interneurons, again, are not coming or going. They're not bringing information in or sending messages out. They're communicating within the central nervous system. And so most of these are located in the brain and the spinal cord, and they work between sensory and motor. So sometimes they can communicate between a sensory and motor neuron, and they can distribute sensory information or coordinate a more complex motor activity, but also can be involved in higher functioning activities like memory and planning and learning, things like that. And so these are very significant. They outnumber all types of neurons combined. So these interneurons play a really significant role in the complexity of our central nervous system. All right, so that's neurons. So we talked about the different types of neurons and the structure of the neuron and how it works and all that kind of stuff. That's one cell type we're going to see in nervous tissue. As I said, there's another type, neuroglia. Neuroglia, or glial cells, do not communicate messages or send messages to targets throughout the body. Their role strictly is in supporting, right? As we're going to find out, the central nervous system and peripheral nerves to a certain degree, but especially the central nervous system, very delicate tissue, cannot survive in the environment of our, just interstitial fluid. It needs to be protected and separated from that. And so neuroglia play a really important role in protecting and supporting these neurons so they can only worry about one thing, and that is sending messages, communicating messages. So these supporting cells are really important from that standpoint. They're half the volume of the nervous system, and there are many different types that we're going to talk about. There are 4 in the central nervous system we want to look at, and there are 2 in the peripheral nervous system. So the 4 types of glial cells in the central nervous system are epididymal, astrocytes, oligodendrocytes, and microglia. Okay? First type is an epididymal cell. Epididymal cells form a type of epithelium called epidyma, which essentially, these epididymal cells are found lining inner chambers within the spinal cord and within the brain. We call those chambers the central canal of the spinal cord and the ventricles of the brain. And what these epididymal cells do is produce the fluid that resides within these chambers. That fluid is called CSF, or cerebrospinal fluid. Oftentimes these epididymal cells have cilia or microvilli to help circulate and move the CSF. They can monitor the CSF and just we'll talk about CSF a little bit later, but it's this really important fluid, cerebral spinal fluid, found in the brain and the spinal cord, that's really important to the overall health and protection of the central nervous system. So that CSF is coming from these ependymal cells, right? So if you look at the image, you can see here is one of the 4 categories of ependymal cells. And this is a great slide because it sort of shows a picture of it, gives you an idea of what it's doing, lining the ventricles and the central canal, assisted producing, circulating, monitoring of CSF. All right? So that's one type, ependymal cells. Next are what are called astrocytes. And astrocytes are these larger cells, as you can see in this image, that help maintain the blood-brain barrier. We'll talk about the blood-brain barrier, but basically it's this barrier that separates out the general circulation from what's allowed to cross over into the central nervous tissue. Provide structural support, can regulate nutrient concentrations, gas concentrations, recycle neurotransmitters, so a lot of functionality from these astrocytes, right? And so that's what we're seeing in the second category of central nervous system glial cells. Third type, and here's a nice shot also of an ependymal cell. You can see this is the central canal of the spinal cord here. If you look over here, you can see here's where all the CSF is going, and it's produced by these ependymal cells, which you can see right there. Okay? All right. So, next type of neural cell is what's called an oligodendrocyte. Oligodendrocytes are responsible for myelination in the central nervous system. You may be familiar with myelin. It seems like most students, when I ask them about this, they at least have some understanding of what myelin does. But myelin is a sort of a lipid-based sheath that insulates and surrounds axons so that impulses, or action potentials, right, that are generated by the neurons move very quickly or move very fast, increases the speed of the movement of these action potentials along the axons. So myelination comes from these oligodendrocytes. And so as you can see in this image, here's an oligodendrocyte, and you can see it's forming myelin. Okay? So myelination is a really important concept. We're going to talk about a little bit more when we look at conduction of impulses when we do neurophysiology. But really important structurally and functionally, obviously, in terms of increasing speed of conduction of impulses. Now, when an axon is myelinated, it's not, the myelin sheath doesn't run the entire length of the axon. I mean, it does run the length of the axon, but it's actually broken up into segments, so it's not one big long segment. And those segments are called internodes. And so the parts that are myelinated are called nodes. And they also call those nodes of Ranvier. And these are basically the gaps between the internodes. And so if you look at this image, you can see here are the nodes, right? And so when you talk about impulse moving on a myelinated axon, the impulse skips the sections that are myelinated and is thought to jump from node to node in this manner. And so it moves much faster because it skips those sections and basically only depolarizes at the nodes and skips the insulated section. So there you can see a nice section of the myelin covering the axon, and it's a great image to understand that. All right. So again, as I said, gray matter is going to be your darker neuron cell bodies and sometimes unmyelinated areas, but the white matter, which is also going to be pretty significant in terms of what we'll see when you look at the nervous tissue in cross-section, are going to be where you have myelinated axons, okay? And that's where white matter comes from. So gray matter, white matter coming from those. All right, so oligodendrocytes then are responsible for myelination in the central nervous system. There is another type of cell that's responsible for myelination in the peripheral nervous system, and we'll talk about that in a little bit. So the last type of cell then is what's known as microglia, or these microglial cells, which have the ability to migrate through the neural tissue and, again, clean up debris and waste and pathogens and things like that. So they're sort of a defense type of cell. And you can see that in this previous image right here. Like here's one right here, microglial cell. All right? All right, so those are the 4 types of glial cells in the central nervous system. Okay? There are 2 types of glial cells in the peripheral nervous system. Okay? We have what are called satellite cells, and we have what are called Schwann cells. Okay? Satellite cells surround neuron cell bodies and help regulate oxygen and CO2 and nutrient content. And then Schwann cells are responsible for myelination in the peripheral nervous system. So same concept as we saw with oligodendrocytes, except there is a different type of cell that produces myelination in peripheral axons, and that is known as Schwann cells. So there's 4 types of glial cells in the central nervous system, 2 in the peripheral nervous system, and those the 2 in the peripheral nervous system are satellite cells and Schwann cells. All right. So to summarize, right, the difference between neurons and neuroglia: neurons perform all the communication, information processing, control functions of the nervous system, where the glial cells, or neuroglial cells, preserve the structure and the integrity and the physical and biochemical integrity of the nervous tissue. So the glial cells are essential, right? They are essential to survival and function of neurons. Oftentimes you have a disease state or a neurodegenerative state. Researchers might look towards glial cells to see if they, for some reason, are being inhibited from doing their job and providing adequate protection for the neurons. So the glial cells are really important from that standpoint. All right, so that gives us, at this point now, we've looked at the overall just sort of organization of the nervous system and how it's organized. Then we looked at neurons and glial cells specifically, how those work and how they're organized. And now we can move into our primary physiological concept for this chapter, and that is neurophysiology. And so neurophysiology, kind of like we did with muscle physiology last time, is really sort of taking it step by step and looking in great detail how these impulses are generated, how they're conducted along an axon, what happens when they arrive at the target, and what effect can they have at targets that they're trying to communicate messages with. Okay? And so there are 5 basic steps or segments of neurophysiology. It's called resting potential, graded potential, action potential, synaptic activity, and then information processing. All right? And this is a great slide sort of showing the progression of how this is going to work. We're going to start with what's happening at rest. And if you have a stimulus of some kind, that will lead to a graded potential, which then may produce an action potential, which can be conducted along the axon and arrive at the synapse, which will then communicate a message and cause information processing to occur at the postsynaptic target. Right? So you have resting, graded, action, synaptic, and information processing. So we're going to look at each one of those components as we sort of go through this. All right? All right, so the first thing, and again, this is sort of summarizes resting, graded potential, action potential, synaptic activity, and then finally information processing. So if you read each one of those, the resting potential is the actual electrical potential across the neuron cell at rest. Graded potential is a temporary localized change in that resting potential, typically a depolarization caused by a stimulus. An action potential then is an actual electrical impulse that propagates along an axon to a target or a synapse. Synapse is where the neurotransmitter is released and the message is communicated. And then finally, the information processing is the response from that postsynaptic cell or that target. All right? So we want to start with transmembrane potential or resting potential. That's the first concept. Okay? And the first things you want to understand about resting potential is that the extracellular fluid and the intracellular fluid differ in ions. So that means the amount of positively charged ions you have outside the cell versus inside the neuron cell are going to be different. And so oftentimes when you have this difference in ions between these 2 locations, inside the cell and outside the cell, you're going to get a charge across that membrane. That's what we mean by transmembrane potential. Okay? Typically, cells have selectively permeable membranes, and that's what creates this charge across the membrane. Okay? And so all cell membranes can produce electrical signals by ion movements. We saw that when we did skeletal muscle, right? When the neurotransmitter acetylcholine bound to the skeletal muscle membrane, right? We said that open channels along that membrane allowed ions, positively charged ions, to move into the cell, and that resulted in a depolarization of that skeletal muscle membrane. Okay? Same thing can happen at the nervous system. Okay? At rest, the inner surface of the cell membrane is negatively charged, typically to negative 70 millivolts, okay, with respect to the exterior. Okay? This is caused primarily because you have more positively charged sodium ions outside of the cell than inside the cell. And if you have a positive and negative ions being held apart, you create this potential energy at that site. And so the resting potential is sort of like coiled spring or water behind a dam, and the cell membrane is acting like that, blocking that dam across the river, right? Or across that water body. And so if you have an opening anywhere, right, the ions can move across. And so typically what we're going to see is a resting potential of negative 70 millivolts. And now a way to generate an impulse of some kind is to change this negative 70. And typically what's going to happen is that negative 70 is going to become more positive, right? And when the charge becomes more positive, that is what we call a depolarization. Okay? All right, so that's what we want to look at as we talk about this topic. So there are active forces across this membrane. The sodium ATPase pump or exchange pump powered by ATP really pumps sodium ions out of the cell and brings 2 potassium ions in the cell. And so as a result, you get this maintaining of this negative 70 millivolts because you're getting a lot more positive ions being pumped out of the cell. So this slide sort of shows what we're talking about here. This is outside the cell, right? This is inside the cell, and as you can see, the voltage is showing a more negative charge inside with respect to the outside because you've got all these positively charged sodium ions outside of the cell. And so as a result, you get a more negative charge inside. Now, if I'm telling you that this negative 70 charge across this membrane, the way to cause a depolarization is to make it more positive, how do you think that can happen? Well, what would happen if we somehow got positively charged ions in the cell? And you see how that charge would then change and become more positive. So that's really the trick. Somehow we need to get all these, not all of them, but these positive ions that are being held outside of the cell, somehow we have to get them inside the cell. You get more positive ions inside the cell, that's going to be a depolarization, and that's what's going to generate the impulse. Okay? So these membranes have sodium and potassium channels, and they're either passive or active. Passive channels, also referred to as leak channels, are always open, and that's what helps establish this resting potential or this negative 70 millivolts, right? Passive channels help with that. But we also have these things called active channels or gated channels. And these are closed at resting potential, but they will open if you have some sort of stimulus. Okay? And so that's what we want to look at, are these active channels, because if these active channels can open, then you're going to allow these positively charged ions in the cell, which will result in a depolarization, which is making that negative 70 a more positive charge. Okay? So a change in transmembrane potential results from the opening or closing of these active or gated membrane channels. And there are 3 ways you can get these channels open. Either a chemical stimulus of some kind, a change in voltage, and a mechanical stimulus. If you have one of those 3 things happening, right, you're going to get these voltage gates to open, and then you're going to see a change in transmembrane potential. All right, so we'll look at each one of these. The first type is what's called a chemically gated channel. The chemically gated channel, again, will be closed at rest, but it will open in the presence of a specific chemical. The chemical you should be thinking of is a neurotransmitter, like acetylcholine. Right? Acetylcholine binds to the cell membrane, the cell gates open, and sodium ions can then flood into the cell. Okay? Found only neuron cell bodies and dendrites and neuromuscular junction is a great example of that. We already talked about that, right? That's how that one works. Okay? So, chemically gated channel, right? It's going to open or close in response to the release of a chemical like a neurotransmitter. Voltage gated channels are channels that will open in response to changes in transmembrane potential. Okay? They have activation and inactivation gates. And this is what we see in an excitable membrane when an impulse is trying to be conducted along a membrane. As voltage changes at one point, sort of like a domino effect, it then opens adjacent voltage-gated channels. When those open, more positive ions rush into the cell, the voltage changes again, and the next segment moves on. So it's sort of like a domino effect. Once you get one voltage membrane channel changing, it opens the next, which opens the next, and that allows the impulse to move. So this is what we see in neural axons, skeletal muscle, sarcolemma, cardiac muscle, utilize these voltage-gated channels. And then finally, mechanically gated channels are channels that will open in response to actual physical distortion of that membrane. So for instance, like vibration in our fingertips, right? The reason you can sense that is because it's the physical stimulus that stimulates that nerve ending, and that is what opens the channels and causes depolarization, which sends the impulse into the central nervous system. And so we have some nice images here where you can see each of these at work, right? Here is a chemically gated channel. First, you can see outside the cell, inside the cell, right? And then the gate is closed, right? The positively charged ion cannot get in. But if you have a chemical of some kind, like ACH, right, binds to its receptor. When it binds, that opens the channel, and as you can see, positive ions come into the cell, right? So that is a chemically gated channel. Voltage gated channel, as I said, is going to change when voltage changes. So you have negative 70. Let's say upstream the voltage has changed now to negative 60. That opens the gate. Positive ions flow into the cell, and the impulse moves on. And then finally, mechanically gated channel, right? The channel is closed at rest. That gives you keeping all these positive ions out, establishing your negative 70. But if you have some sort of stimulus, like again, like vibration or touch, hearing is also a mechanical stimulus. As sound waves, we'll talk about that when we do hearing coming up. But whatever, any mechanical stimulus, these will respond to that, open the gate or the channel, and our positive ions will move in, and you get a depolarization. All right. So the trick is to get positive ions in the cell so it depolarizes, right? Because it's at negative 70 because all these positive ions are being held out and resulting in a more negative charge inside the cell. But to get something to happen, we need to make that charge more positive. How do we do that? We need to find a way to get positive ions in the cell. And those are the three ways it can happen. Okay? All right, so that's resting potential. So make sure you have a good understanding of that. If we see a depolarization, it's going to initially cause what's called a graded potential, also referred to as local potential. And these are changes in transmembrane potential that cannot spread far from that site of stimulation. Okay? And so any stimulus that opens a channel is going to result in a graded potential because that's what depolarization is. Okay? Hyperpolarization can also happen, which is becoming more negative, right? So going from negative 70 to negative 80 perhaps. And then repolarization is also very important because once it depolarizes, it needs to get back to resting state so that it can sort of do it again, right? Be able to do that again. So graded potential, you have resting negative 70. Let's say there's a chemical that opens those channels. Transmembrane potential goes up, and that results in a depolarization. All right? So you have negative 70 at rest. You get depolarization, which is, again, a shift in transmembrane potential towards 0 millivolts. Typically because the movement is positively charged sodium ions through the channel, and that depolarizes the nearby plasma membrane, and the change in potential is proportional to the stimulus. So if you don't have a really large stimulus, you're not going to get that much change in voltage. So for instance, in a graded potential, you might go from negative 70 to, let's say, negative 65. Okay? So you can see it's not a big change in voltage because, again, it's proportional to the stimulus. The smaller the stimulus, the less significant change you're going to have. So you might get change in negative 65, but it's not going to spread that far because it hasn't reached what we call threshold yet. Okay, and so when the stimulus is removed, we're going to see repolarization, which is returning back to -70. And I already mentioned hyperpolarization can happen as well, which is increasing the negativity of that resting potential. All right, so that is a graded potential. If you have a greater stimulus then produces a graded potential, it can then result in what we call an action potential. Okay? And an action potential is a potential that's going to be propagated along an axon and affect an entire excitable membrane. Okay? And so that's how an impulse can get moved from the neuron all the way down to the synapse. Okay? So that's what I want to look at first. Right? Initiating an action potential requires a depolarization large enough to reach what we call threshold. And for our purposes, we're going to say threshold is -60 millivolts. Okay? So that means in order to generate an action potential, you need to go from -70 all the way to -60. Right? That is what we call threshold. I'm just going to write, if I can, threshold. I'm going to write that well, but it does the trick. Okay? Anything before that would be a graded potential, as we already talked about. Right? Anything that reaches threshold, you now have an action potential. Okay? Make sense? All right, so it needs to change 10 millivolts to reach threshold, and then you'll have what we call an action potential. And we call this the all-or-none principle, because if the stimulus exceeds that threshold amount, the action potential will happen. So the action potential is either triggered or it's not, all or none. All right, and so you can see in these images, start with -70 at rest, right? You depolarize the threshold, okay? Once you depolarize the threshold, you're going to see activation of more sodium channels, which sodium ions are going to rush into the cell. Intermembrane becomes more positive, and you have this rapid depolarization, very significant. The impulse is sent typically at around +30. The inactivation gates will close, and repolarization will begin. And then finally return back to normal permeability. You may have some hyperpolarization, but the action potential is over at that point. All right? All right, and so there is a refractory period during which time the membrane will not normally respond to additional stimuli. Let's see, I don't think I'm going to go over absolute and relative refractory period. Let's move on to propagation then of action potential, right? So once you reach threshold, you need to then propagate that action potential to the target. Okay, so that's the movement of the action potential along the entire length of the axon. And there are two ways in which an action potential can be propagated. You have what's called continuous propagation and saltatory propagation. Continuous propagation is conduction of an action potential along an unmyelinated axon. Okay? Saltatory propagation is conduction of an impulse along a myelinated axon. And as we talked about before with myelination, the big difference is speed. Right? Continuous propagation on an unmyelinated axon is obviously going to take longer. Saltatory propagation is going to be faster. All right? So, continuous propagation is, again, the propagation of an action potential on an unmyelinated axon, and affects one segment of the axon at a time. So if this is a neuron cell body here, and you have an axon, right, that's not myelinated here, and it ends at the telodendria. This isn't the greatest picture in the world, but hopefully you're going to get the idea. Okay, that impulse is going to have to depolarize one step at a time, all the entire way along the axon, because it can't skip anything, right? When we say with myelinated axon, you can basically skip the myelinated sections. With unmyelinated axon, there's no myelin, so it has to depolarize every step along the way, and so it just takes more time, right? It's slower. Now, again, slowness is a relative term. It's still fast, but not compared to myelinated axon, which is much faster. So that's continuous propagation. Saltatory propagation is a conduction of an action potential along a myelinated axon. And this is much faster, uses less energy, because the myelin insulates the axon, essentially preventing what I just described, continuous propagation. The impulse will jump from node to node, and depolarization only occurs at those spaces where there is no myelin. So it goes much faster. Okay? So that's saltatory propagation versus continuous propagation. And this image sort of shows that, right? You have a depolarization here, and it's looking for the next place to depolarize, but it can't because there's myelin right there, right? So it's going to go to the next available segment, which is the node. It will depolarize there, right? And local current moves the ions to the next component, the threshold, big depolarization, and the next thing moves on to the next spot, right? So anyway, it moves much faster. Okay? And so this myelin is really important. We're going to see myelin conducted to things that need really fast impulses, like skeletal muscle and reflex pathways, right? Our vision, these all things need myelinated axon. And so there also are demyelination disorders where the myelination becomes damaged. And a great example of that is multiple sclerosis, or MS, which I'm sure you're all at least somewhat familiar with. This is a chronic, potentially debilitating disease that affects the central nervous system. It's thought to be primarily an autoimmune disease because the body is essentially incorrectly directing antibodies and T cells and things like that to attack the myelin sheath. Instead of attacking pathogens and other things that it's supposed to attack, it starts attacking the myelin and causes damage. So in multiple sclerosis, the body incorrectly directs antibodies and white blood cells against the myelin sheath. This causes inflammation and injury to the sheath, and then ultimately hits that nerve ending at that location. So it results in multiple areas of scarring, hence the term multiple sclerosis. And so this damage over time can cause impulses to be slowed significantly or blocked altogether, and often things like muscle coordination, strength, sensation, vision can all be impacted because these are the types of impulses that travel on myelinated axons. And so this is sort of an image showing you generally, right, normal healthy cells going to jump from node to node and arrive at the target. And here you can see in multiple sclerosis, right, the T cells and the antibodies are targeting the myelin sheath and destroying it and attacking it, and so it's going to be harder for impulses to get through to the target. All right? So that is a demyelination disorder. All right, so these axons obviously are going to play a really important role in propagation of an impulse, right? And so diameter and myelination are really the two factors that impact how fast an impulse will travel, right? And so you have three types of axons, type A, B, and C, and they're, again, basically classified based on whether or not how large their diameter is. As larger diameter axons are going to move impulses faster. Myelination, of course, and then ultimately that will impact the speed of the impulse. So type A fibers are myelinated and have a pretty large diameter. So obviously those are the ones that are going to move the fastest, right? Very, very rapid information to and from the central nervous system. So these are the types of things that will be attacked primarily on multiple sclerosis we just talked about, right? So position, balance, touch, motor impulses, coordination, all those things go on type A myelinated fibers. Type B fibers are also myelinated, maybe not quite as large in diameter, and they're still going to send information pretty quick, but it's a little bit slower than type A fibers. So sensory information, peripheral effectors can have type B, and then type C fibers are unmyelinated fibers and have a smaller diameter, so the speed is much slower, right? This would innervate, like smooth muscle, involuntary smooth muscle and gland control, right? The fact that when you start to sweat, it doesn't matter that it happens lightning quick, right? Just as long as the impulse gets there. Same with smooth muscle, right? The impulse it's not necessary, not essential that the impulse travel incredibly fast. So these are going to be unmyelinated, smaller diameter fibers, all right? So information travels within the nervous system as propagated electrical impulses or action potentials. Most important information like vision, balance, motor commands, these are carried by larger diameter myelinated axons, which are type A, right? All right, so next part then, after the action potential, is the synapse, right? That's where the impulse will arrive. Okay? And so the synapse is the site of communication between a nerve cell and some other type of cell. Now this last part here, this coming part with the synapses, we've sort of done this already with neuromuscular junction. So this will kind of be review at this point. But the synapse is communication between the nerve cell and the target. And actually there are two types of synapses. We've looked only so far at chemical synapses, and that's what we'll continue to focus on. But there is this other type called electrical synapse, which actually has direct physical contact between the cells, but that's pretty rare. Chemical synapses are very common, and that's what we saw, for instance, at the neuromuscular junction. Right? A chemical was released, a neurotransmitter moved across this gap, this synaptic cleft, to bind at the target. Okay? So there is this electrical synapses. Again, these are pretty rare. What we want to look at are chemical synapses, right? Chemical synapses are synapses between neurons and other cells, right? Cells are not in direct contact and they must use a chemical, so they use neurotransmitters. And there are different types of neurotransmitters, hence there are different types of synapses. So a synapse that uses acetylcholine, we're going to call that a cholinergic synapse, right? Like dopamine is not going to work at a cholinergic synapse, for instance, right? It needs to be specific to that type of neurotransmitter, okay? So cholinergic synapse is any synapse that releases ACH. So some examples are neuromuscular junction, which we already talked about last time, many synapses in the central and peripheral nervous system, and then some like glandular junctions in the parasympathetic division. We'll talk about those when we do the autonomic nervous system. This part is pretty much review the events at a cholinergic synapse, just to make sure you have a good understanding of it. The action potential arrives and depolarizes the synaptic terminal. That causes calcium ions to enter in and trigger exocytosis or release of acetylcholine out into the synaptic cleft. Synaptic cleft, the ACH now will bind to its receptor, depolarizing the target, and then the ACH or acetylcholinesterase will remove and break down the ACH into its components, acetate and choline. You can see that in these slides following. Right? So you have the action potential arriving, right, causing exocytosis or release of the neurotransmitter. The neurotransmitter binds to its receptor in the postsynaptic membrane. That depolarizes the postsynaptic membrane and sends an impulse that will be propagated at that target, and ACH will also be removed and degraded by the acetylcholinesterase or ACH. All right? So there is a synaptic delay across these synapses because you need time for this action potential to arrive and have its effect at the target. Okay? The significance of this delay is that fewer synapses means a faster response. And so we're going to be looking at reflexes in the next chapter when we look at spinal cord. And some reflexes may only involve one synapse. We call those monosynaptic, and those are the fastest reflexes you can have. But there are other types of reflexes that have multiple synapses that maybe take a little bit longer, but give you a more complex response that can occur, and we call those polysynaptic. So this synaptic delay is significant, as we'll see when we look at reflexes. All right, so there are other neurotransmitters in the body, as I said, norepinephrine, dopamine, serotonin. We've talked about ACH already. Norepinephrine is released at adrenergic synapse, typically has an excitatory and depolarizing effect, widely used via the autonomic, especially sympathetic. This is like our adrenaline hormone that is released and communicate messages. Dopamine is another one. May be familiar with dopamine from its association with Parkinson's disease. We'll talk more about that coming up when we look at the brain. Serotonin is another one you may have heard of that impacts emotion and mood and attention, right? So a lot of drugs have like serotonin receptor uptake, promotion, things like that, to really try to, increase the effect that these, sort of feel-good chemicals can have for our brain. But a lot of the drugs out there that impact the nervous system sort of try to manipulate this neurotransmitter system. But sort of the downside, if you will, is that it can side effects. What other types of things could potentially happen in terms of emotion and motor control, perception, other things like that. So those can be tricky to kind of work with. So those are different types of neurotransmitters. We'll talk a lot about neurotransmitters coming up. There also are these things called neuromodulators, which sort of modify the effect of neurotransmitters. Typically, a great example of a neuromodulator is an endorphin. You may have heard of endorphins sort of associated with exercise, right? Like that sort of runner's high, or that just feeling of invincibility you get when you exercise sometimes, is thought to be because endorphins can be released when you have higher levels of like cardiovascular exercise, for instance. And basically what endorphins are thought to do is to modify our response by sort of limiting these pain chemicals like neurotransmitters, like substance P, for instance, is a neurotransmitter that transmits pain messages to the brain. So if you can have a neuromodulator that's going to modify that by like slowing that or blocking or blunting the effect of that message that's telling us this exercise is painful, then maybe you're able to keep going and sort of feel no pain, right? I feel no pain at this point. So endorphins are known to do that as they associate with exercise. All right, so the last part of this neurophysiology then is information processing, right? What happens at the target once the message is received, right? And so typically, and what we've seen is, once that neurotransmitter binds to the target, has an excitatory response and causes something to happen, like we saw with skeletal muscle, right? We saw acetylcholine being released, it bound to the sarcolemma, and that ultimately led to the muscle to contract, right? And so that is an excitatory response. But not everything is excitatory. Sometimes you can have an inhibitory effect as well. And so that's what we want to look at here in information processing, okay? And so once the impulse is generated in the postsynaptic cell, you have what then are called postsynaptic potentials, right? And postsynaptic potentials can either be excitatory or inhibitory. An excitatory postsynaptic potential is one that's going to cause a depolarization of the target. An inhibitory postsynaptic potential is going to cause a hyperpolarization, right? Going away from positive, right? So maybe negative 70 to negative 80 it might cause, and that way there's going to be no way to reach threshold, okay? So excitatory is going to depolarize, inhibitory is going to hyperpolarize, right? So if a neuron receives many inhibitory postsynaptic potential, it's not going to produce an action potential, right? Because it's not going to reach threshold, okay? But when you have an excitatory response, you typically need more than just one excitatory postsynaptic potential. And so to do that, you need to sum up the effects of multiple excitatory postsynaptic potentials, or EPSPs. Okay? And when EPSPs combine, we call this summation. And there are two types of summation that you want to know: temporal summation and what we call spatial summation. Temporal summation is where you have one synapse firing repeatedly, right? And you add up the effect of those multiple impulses, and you reach threshold. Okay? Spatial summation is where maybe you have multiple locations, and those stimuli are arriving at the same time, and you can add up those impulses, right? And so these temporal summation, as you can see, the first stimulus arrives. That is going to maybe get a graded depolarization, but that's not going to generate an action potential, right? Because you need to reach threshold. So then a second stimulus arrives, and it's added to the first stimulus, then you get the threshold, and then an action potential is generated. So typically one EPSP is not going to be enough. So you have to have multiples. And when you have multiples, you're summing up their effect, and that's where the term summation is coming from. Spatial summation, same concept, it's just you have more than one site where this is occurring. So you have one impulse coming from one, another from another, and you add up their effects collectively. They cause threshold to be reached, and then an action potential to be sensed. All right? All right, so that's it. Don't worry about facilitation. These last few slides are really just associated with sort of just summarizing what we've talked about. So if you want to read through those, you're welcome to do so. But that's it for the nervous tissue and neurophysiology lecture. Next one to be posted will be the spinal cord. Hope to get that up soon, and then after that we'll do the brain. All right?